So you can start with the introduction and ask the resource person to share the screen.
Madam, please share your screen, madam. Please give slideshow, madam.
Ma'am, please share the entire screen rather than the window. Mali from just talk to her.
Madam, you are not audible.
Can you unmute yourself?
Yes.
Madam, you just
Share the
you select the entire screen, madam.
Okay.
First you unshare this one, madam. Then again you share. Madam.
Just 1 second. I'll just close it again and then.
Okay. You go to that button, that one. Madam, present now. You click on that, madam, below your screen. Present now. Then you select that entire screen. Madam. Sir.
You have to click on the screen being shown at the center.
No, earlier it was coming.
Yes, I understand, ma'am. Can you please follow my instructions?
Yeah, please.
Once you click the entire screen, there would be a screen displayed in the middle. You have to click on it so that the share button would be enabled.
No. I am in that page only, but the PPT is not visible.
No, you have not shared yet.
No issue, madam. Just you click on that entire screen, madam. Automatically it will come. First you open the madam, first you open your PowerPoint, madam.
No problem. First click on the and
No,
share
everything is open, Dr. Manjan Gowda. I am in that share
Yes,
button
madam.
only, but I am not able to click the PPT for sharing.
Yes. I am telling you, at the middle of the screen, there is the screen shown. You have to click on it first. So that
Yes, sir, I know that. Yeah, but it's not coming. PPT is not visible in that screen to select it.
I know. Share
Just
the
one.
entire screen anyway. Share the screen anyway. Then you can switch. Share entire screen.
Yeah, 1 second.
Share your entire screen. Then the screen would be shown in the middle. Click on it and press share. Yes. Now at the bottom, go to PowerPoint. We are seeing everything on your screen. Go to PowerPoint in your system tray, where you have PowerPoint open at the bottom.
Yes, madam.
Okay.
Go to slideshow. Go to PowerPoint and go to slideshow.
Slideshow mein
Is it visible
perfectly
now?
fine, madam.
Yes.
Yes,
Okay,
madam.
okay.
Click on the hide
madam,
option
yes.
only. Click on the hide option only. Hide.
Okay.
Click on hide.
Good morning.
Hide.
Option, madam. Hide. Okay,
Yeah.
fine. Perfectly fine, madam.
Okay,
Okay,
thank
good
you.
morning, dear participants. Good morning. I welcome you all for this session. I introduce the today's speaker, Dr. Rindita, madam, presently a senior scientist at our regional station in the discipline of entomology. And madam is having around 13 years of research experience in the field of entomology. Before joining our regional station, she has worked nearly around 10 years at ICR Central Citrus Research Institute. Madam, I welcome you for this session. Madam, kindly start your presentation.
Yeah. Thank you, Dr. Manjan Gowda and other training coordinators. And I welcome all the participants who are online to this presentation titled Modern Tools for the Management of Seed Infesting Insect Pest. And I think yesterday, Dr. Revanasita might have already given you an exposure for the management, safe storage of the seeds as well as the grains. So if any portion you feel it is like repetition of yesterday's class, kindly let me know. I'll just skip those slides also. So now coming to the presentation. like see, our institute is National Institute of Seed Science and Technology, and you all know that our prime mandate is, or our focus is on seeds, only seeds. So I'll be always, my presentation will be always revolving around seeds and not grains. So I think I need not tell you what is the difference between the seed and the grain. So coming to the seed production program, whenever a farmer is involved in the quality seed production program, the quality of the seeds is of high importance. Why? Because if the seed that you are using for sowing is good, means the farmer will get a good crop or high yield in the coming season. So if the seed is already damaged due to biotic or abiotic factors, then that is a question mark. So we have to see what are all the means or the ways to manage these issues. So I'll be focusing only on the insect pest side. And that literature says that almost in a year, 10 to 40% losses are losses of food grains are caused exclusively due to the insect pest infestation. And of this, about 20 to 40% losses happen during the post-harvest period. So just imagine that the farmer is harvesting the crops, and he will be shifting the harvested produce to his go down or the storage place wherever the harvested produce is stored. There he'll be cleaning the seeds. The seeds will be cleaned, and sometimes it will be processed. Sometimes it will be treated with some fungicides or some insecticides or any growth regulators. And he will be storing these seeds mostly until the next sowing season. So this until the next sowing season is a period. It could be 3 months or 6 months or 9 months or even 1 year, depending upon the type of the crop or this sowing season. So during this storage period, mainly the culprits like insect pest and the fungal pathogens, and apart from that, other rodents, birds are all come into the picture, and they cause the damage. And in India alone, more than 100 or almost 111 insect species have been reported, which causes damage to the seeds. So when I say causing damage, here it is not only during storage, it includes both those insects which starts their infestation from the field. For example, we can classify these storage insect pest into two groups. That is, as I have mentioned in the slide, that's primary storage pest as well as the secondary storage pest. So the primary storage pest, as the name indicates, it will feed on the boiled seeds or the freshly harvested produce. And the secondary storage pest, they don't have a strong mandible or a mouth part, so they will be feeding on the broken grains which has been left over by this primary storage pest. And this primary storage pest can be further grouped into internal feeders. That's the external feeders. I think I need not explain the details. It's understood. So just some photos I want to show you. What are the major insect pests of seeds under storage? So the rice weevil, the lesser grain borer or Rhizoperta dominica, some of the moths like Citotroga, Cerealella, then cigarette beetle, drugstore beetle. Here you can see the damage of a rice weevil as well as the lesser grain borer. See here, this is the damaged wheat seed, and here on the right side is the healthy one. So the entire endosperm, embryo, everything has been eaten away by this Rhizoperta, lesser grain borer adult, and the intermediate stages. Here you can see the pulse beetle, which is very common to almost all the pulses, or you can say the legumes. So this is a cut open chickpea seed, and here you can see the different larval stages of this pulse beetle. And on this is a seed coat of the chickpea where you can see the eggs laid, and this is the larva of pulse beetle. And this is just a small short video. Just I wanted to show you the damage by rice weevil on maize seed. See, it was an intact and very good maize seed only, but you can see the kind of damage it has caused, and later on, due to this infestation, it will be converted to mere powder by these insects. And here, this is how much is the extent of damage caused by this pulse beetle larva. See, here you can see all the beetle And this is I'm removing it from the chickpea seed. And this is not from many seeds. This is from a single seed I have collected. And even I have recorded up to 28 number of larvae within a single seed. So you can imagine the kind of damage these. So this is again some of the secondary pests like red flower beetle. Red flower beetle is very popular among the storage pests because highest insecticide resistance have been reported in the case of red flower beetle, and it is very difficult to manage this red flower beetle. And down is the sawtooth, the grain beetle, and here you can see the infested garlic bulbs, and it is damaged due to the almond moth. And this is rice moth, which is very common. So if rice moth, this is even in your grains also at home, your household items also, you may see this. If the webbing is observed, means you make sure that rice moth infestation is there. And this is the capra beetle. So this is just some of the photos I've taken. So what is the significance of the storage pest? And you all know that it leads to the qualitative as well as the quantitative losses. Just to cite one example, it is estimated that a single paddy kernel weight is almost 20 milligram, and one single rice weevil, for completion of its life cycle, it will feed almost, it will consume almost 70% of the weight of the seed. And the single female of this rice weevil, in her one generation time, she can produce almost 5 lakhs of offsprings, and this 5 lakhs of offsprings will almost consume the rice quantity amounting to almost 10 kg. So you can imagine, when you see it is just a single rice weevil, but you imagine within a short period of time of like 1 month or 2 months, the progeny will be multiplying at a faster alarming rate, and the coming progenies will be consuming the seeds, and you will not come to know, maybe after 2-3 months when you open the bins or your seed bags, only you will come to know that the infestation levels has gone to an alarming rate. So that is the kind of damage done by this storage insect pest. So apart from this qualitative and quantitative losses, some of the parameters as a seed person we look into is, one is the seed germination. So if the embryo is intact and endosperm is also there, so it will result in a good seedling. But if embryo has been already damaged, then there will be no seed germination. And apart from this affecting the seed germination, there will be changes in the pH level as well as the protein content. There will be elevated moisture content, and free fatty acid content also will be varying. And maybe you might have observed, like after infestation by this insect pest, especially in paddy or something, like stale or offensive smell will be there. That is due to the various processes that is happening after the infestation by this insect pest. And when the infestation levels are too high, your seeds will be, you can see that a large number of webbings will be there. Apart from that, body parts like their excreta will be there. If it is a moth infestation, means scales will be there, body parts will be there, and foul smell will be emanating from this bins. So that the entire seed lot will have to reject because it has been already damaged by this. And apart from this, the secondary problem arising is the fungal infestation, additive effect. So the insect damage plus this fungal infestation will completely damage your seeds. So we have to just note down some of the main factors which either you can say favor or that we can manipulate in order to control these insect pest under storage. So that is storage temperature as well as the aeration. And the insect pests are mostly thermophilic, and they prefer an optimum temperature range between 25 to 35 degrees Celsius. But still, they can survive from like 10 to 15 to up to 42 degrees Celsius. So we can manipulate, like we can either treat the seeds with a higher temperature, but in the case of seeds, we have a limitation because it should not cross above 50. If it crosses 50 degrees Celsius, then the seed will be killed. So if it is a grain, you don't have that issue because it's already dead. So you can treat to any kind of like temperature and the temperature period also. So there is nothing to worry. But with respect to seed, we have to see that we don't expose to too high temperatures. So just to cite one example is here we can see the granary weevil, and this table shows the number of days which is required to kill this kill these different stages of this insect pest at different temperatures. See, when the temperature is very low, like if it is ranging between 0 to 5 degree Fahrenheit, just we need only 1 day. So, but if you come to with respect to granary weevil, if it at 15 to 20 degree, 20 to 25, so these are the optimum, the temperature range which is preferred by the insects. So we need to exert more number of days. More number of days has to be there in order to kill this insect pest. So it is very difficult to kill these insects during their optimum your preferred temperature range. So we have to manipulate, like either increase or decrease the temperature in order to kill these different stages inside the seeds. And another factor is like the storage moisture, and you can say the seed moisture content. So we have the Indian minimum seed certification standards. According to the IMSC standards, there are like already some seed standards have been set, and for like cereals, pulses, you should store at a particular kind of range only, like that. So this table shows you that information. So when you compare the cereals as well as the pulses, there is a slight difference in that seed moisture content level you have to keep. So mostly the cereals can be the seed moisture content can be up to 12%. So mostly like between 10 to 12%, you should bring down from the moisture content. See, when you are harvesting the seed, it could be 35%, 25%, 20%. It will be varying depending upon the crop seed. So from the time of harvest, then you have to bring down the moisture temperature to particular like 10 to 12% for the cereals and 8 to 9% for the pulses. So that is the minimum seed standard that have been set with respect to moisture content of the seeds. Okay, now we have seen that what are the different insect pests that attack the seeds and what are the factors that govern their survival in the seeds. So when there is an when there control. So before this, we have to take out some of the prophylactic measures. Like when you have harvested the seeds, immediately you are bringing to the storage go down. So there are different phases at which you have to intervene. So that we can minimize the level of infestation in the seeds. So it could be classified into 3 different stages. That is before storage. There are certain precautionary measures you have to take. During storage, what are the things to be done? And before you distribute the seeds, you are using it for sowing purpose. There are some certain parameters we have to see. So this table just shows when you are taking samples, seed samples for checking whether the insect pest infestation is there or not. This is the category. Just simply, I think you all know this. If there are no insect stages, we will write C, means clear. And if it is less than 20 insects per 90 kg, we'll mention few or light infestation is there. And if it is like 50 to 300 insects have been reported from a 90 kg sample, then we will write moderate number of infestation is there. And if it is more than 1,500 per 90 kg of seed sample, we will write very large number of insects are observed. So this is the kind of recording we will do from the seed samples collected at frequent intervals. So how do you detect this insect infestation? So it can be like further divided into two parts. Two phases. Some are like you can see you by your naked eyes that insect infestation is there, and in some cases there will be no symptoms from outside, but the insect stages will be inside. So I'll just give you a brief idea about how to detect all this insect pests. So the basic funda is like visual observation. So just you need a magnifying glass or if possible, though we have to do like by once in 2 weeks or at least Once in a month, we have to inspect the seed lots which has been stored in your godown. So we have to see for any insect stages, like the adults are there, or any webbing is there, or any broken body parts are there. So like that, if any such things are be seen, it means you have to just take care of the seeds. So the second method is like probe sampling and seeding. So it's just a simple thing. It's actually it's a tube which is having a conical shape down here. You can see the white color cap, and this is a steel frame, and it is cylindrical in shape. It is having perforations. So what you have to do is you have to just insert this probe into the middle of your seed bag, and then at the top of this probe should be at the level of your seeds. So you can see that after a few hours or maybe 2-3 days, you can see if the insect infestation is there. The insects will come, enter through these holes, and they will get trapped in this white cap. And this white cap, conical cap, inside it will be having glue. So the insects cannot come back and then escape. That is never possible. So after that, you can just take out this probe, and then you can make it out whether your seed lot is infested or not. The only disadvantage is that it is laborious, because if you have too many bags, then you need to have like a number of probes, and then it is little laborious, but it is an easy method. Third one is light trap. And you all know most of the storage insect pests are nocturnal. It means they will be active in the night, and they will be attracted to light. So we are using this method to attract these insect pests. We are using a UV lamp. So we have to install this. This is a kind of setup you might have seen. This is a light trap that we have to hang like around 1 to 1.5 meter above the ground level. And you can see that once the light is on, after some time, the insect pest, if it is there, it will be slowly moving and getting trapped in this light trap. So the fourth one is sticky traps. It's a simple thing. We use it for the whitefly, most of the sucking pest management in the field crops. So here you can see the crawling insects. Insects are there. In that case, we can just keep it here and there in your storage bottom, and like all those crawling insects will come and attract it to this yellow sticky trap. The fifth one is the pheromone trap. So mostly when you say pheromone trap, it will be two types. That is a sex pheromone as well as the aggregation pheromone. So sex pheromone is like of the same species will come get attracted and get trapped into this pheromone trap. While in the aggregation pheromone, it will be like both males and females will get attracted. So that is the difference between these. So these, suppose some of the insects like rice beetle, the lesser gray borer, pulse beetle, some of the chemicals are known. And in other foreign countries, they have done the research and found that these chemicals, when it is like loaded into a rubber septa or any dispenser, it is attracting the storage pest, and then it will get trapped into this sticky card. And so this is like a simple techniques which can be used for identifying the infestation which is present externally. So how do you know if the infestation is there internally? So there is with the naked eye, it is very difficult to see. See, for example, this is the photo it's a green gram seed which is having the pulse beetle pupa inside. You can see the embryo here, and this is the cut open in the middle. So from outside, if you see, there is no other symptoms, only except for the eggs laid. And if you are not well versed with that, even that you may ignore. Since we are like into the subject, we know that the eggs of pulse beetle is being laid on the seed coat, and we can easily identify it. If it is for a layman, it will be very difficult. So the simple method is like you can cut and see. You can take few seeds and then cut in the middle and see if there is any stages. And when you cut, if it is there, it will be visible like this. Second method is transclusion method. So in this transclusion method, what you have to do is a few seeds, the samples seeds are being boiled in 10% NaOH solution for around 5 to 10 minutes. And once the boiling process is over, you have to wash the seeds. And by that time, it might have become translucent. And if the insect infestation stages are there inside, you can see like something against the blocking. Like suppose if it is a white rice or paddy, you can see the larva or pupa, whatever. You cannot make it out whether it is a larva or pupa, but you can make sure that there is a stage of this insect pest inside the seed. So then just to make double sure of it, you can just cut the seed and then see whether the insect stages are there or not. The third method is flotation method. In flotation method, we are using two different solutions. Then one is the sodium silicate and another is the methyl chloroform, and the specific gravity has been mentioned here. So the sodium silicate is having a specific gravity of 1.16 to 90, and here you can see that plus water. So there will be a separation layer between these two liquids, and always the sodium silicate will remain on the top. So what you have to do is, after mixing the solution, you have to, in the container, you have to put the seeds. So it will be segregated into 3 parts. On the the bottom part, you will get the healthy seeds. And the middle part, you will get the seeds which is having the early stages of the insect pest. And on the top, you will have the damaged seeds. That means it will be having the later stages or almost the adult stages of the insect pest floating on the top. So this is how you can make it out whether the seeds are damaged or not. Then another method is like Burley's funnel. I think you all will be aware of this. The simple principle is we are using a bulb, and here there is a wire mesh, and this is a funnel, and there is a container with alcohol, or even you can use water. So when the bulb is on, due to the heat generated, and you are keeping the seed samples here, if the insect stages are there outside, because of the heat generated, they'll start slowly moving through the funnel and they'll fall into the water or alcohol, whatever solution you are using. So that is how you make it out that insect infestation is there. The another method is uric acid method, and you all know the uric acid is the main component of insect excreta, and indirectly you are making it you're confirming whether insect infestation is there is through the colorimetric method. So when the color changes, you can make it out that insect infestation is there. And another is the staining method. So staining method, actually it's a bit difficult, but where it is advantageous is especially in the case of like a rice weevil, rhizoperta and all, the paddy seed color will be white, and the egg color is also white, and it will be just the eggs will be laid on the surface with the mouth part. The female will make a small cavity and she will deposit all the eggs, and it will be plugged with the frass. So it is very difficult to make out the eggs. So for this purpose, we are using the staining method. So some of the stains you all know, like acid fusion, then gentian violet. So depending upon the color of the, depending upon the stain you are using, you can make it out whether the eggs are there or not. So that is how indirectly you make it out that the insect infestation is there. So here you can see that this is a trivolium casting, that is red flower beetle eggs, which has been stained with acid fusion. It has turned to pink, bright pink color, and here it is a bromocrosol green. So you can make it out that the eggs are there or not. And another method is hidden insect infestation detector. So it's a simple technique. This is a metal, actually it's an inverted triangle shaped metal structure. Through it is open in the middle, and you have to put the seeds, and these two are the rollers, and in between the roller there is an impregnated paper which is having 0.7% ninhydrin acetone solution. So what happens is you have to put the seed sample through this container, and when it reaches here, the rollers will be moving, and the seeds will get crushed in between the rollers. And if any insect stages are there, so the body fluid will be crushed along with the seeds. That body fluid will be reacting with the ninhydrin chemical, and it will result in a purple coloration on this tape. So if there is no purple coloration, means the seeds are free of insect infestation. And if the filter paper is having a purple color, it means there are insect stages present inside the seeds. And just I wanted to, this is not much detail, I want to show modern methods are still available across the globe. Like olfactory based method, that is the solid phase micro extraction method. This is being used in India also. Like conductance based methods, like electrically conductive roller mills. Then odor detection techniques are there, in which we are using in electronic nodes. Just similar to our nose, we are sensing the smell. So we are using just the manipulation of this human nose only. So it will detect the aroma, and it will be translated to what are the kinds of volatiles present in that. So based on that, we can make it out whether the seed lot is having infestation or not. I think I'll explain one example in detail in the coming slide. And some other, like we can also make out the make out, like especially in the case of rice weevil, if you listen very closely to the container or where the bags are stored, the feeding sound will be there. So just like the acoustic method only, so we can make it out whether, so the signals, the sound will be captured, it will be amplified, and we can make it out whether the insect infestation is there or not. So these are some of the methods which are being used in the other countries also for the detection of this insect pest. So coming to the management aspect. So before management, so we need to know, like for field crops, we have the economic threshold level. Same principle, we have the storage insect pest also. But the only thing is that the ETL for storage insect pest is almost zero. Because if you see, like if a farmer is involved in any quality seed production program, he has to follow some of the standards which have been set by the Indian minimum seed certification standards. So in that standards, it is mentioned that if you are carrying out a foundation or certified seed production, then you have to do seed sampling. And in that seed samplings, like the ETL has been kept at 1% for the seeds like bold seeds or big sized seeds like maize and legumes, and it is 0.5% for other small sized seeds. So just to imagine, if you're taking 100 seeds, in that, even if one seed lot is found damaged by insect pest, it means you have to reject that seed lot. So that is the importance of this ETL level. So we have to use all kind of means. It's not like completely depending upon like either technical means or something, but we have to take care at each levels. It's starting from the harvest stage, even before harvest also we have to do, and from all these different stages we have to take care so that the infestation levels by the storage insect pest will be minimum. So we have to first identify from where these insect pests are coming. So I told mostly when you say storage pest, your thinking will be always confined to the storage godown or storage structure wherever the seeds are stored. But it is partially only correct, because in some cases the infestation will start from the field. For example, like pulse beetle, rice weevil, then ankylosing grain moth. All these pests, the infestation or the adults will start their egg-laying process when the seeds mature in the field. So already when you are bringing the seed lot, it will be having the eggs of these insect pests, whatever I have told you. And you will not come to know, because it is very difficult, except in the case of pulse beetle, if you are experienced, you can make it out. But in the other cases, it is very difficult and physically not possible. So then you store these seeds in your godown, and then slowly after a few months when you open it, the infestation will be there. So there it starts. So the infestation starts from the field, and during the transit already the previous season infestation will be there in your godown, inside the walls, inside the cracks, crevices, everything. Their stages will be there. So, and if you are storing the seeds in the previously stored bags, then chances are there that different stages will be there. So it will start infesting the new seed lot also. So all this, it may be like small things, minute things, but we have to take care so that you can prevent to some extent. So first thing we have to do is some minimum things are like we have to do the timely harvesting. After harvesting, you have to properly dry the seeds so that you are bringing the seed moisture content to 10 to 12%, and you have to properly thresh it, clean it, and then store. If it is possible to do some chemical treatments, then you can do that and store the seeds. See some of the like storage bags that have been used the previously many years back. We have been using gunny bags. Now it has improved, and we are using HDPE bags, jute bags, and nowadays PICS bags and hermetic bags is a very popular terms you might have heard of. And coming to this PICS bag, this in mostly in African countries they are storing using this PICS bag for storing the pulse seeds. So here you can see that there are two kinds of bags. From the exterior, one cup bag is there that is made of polypropylene, or it could be nylon material, and inside you can see a transparent bag that will be mostly made of polyethylene bags. So only thing is that once you put the seeds inside, it has to be tightly covered, so that no maximum no air should not be there. So similar kind is another hermetic bag is grain pro bags, which is very popular in the market. So it is airtight, so insects cannot survive inside such bags. So what is the difference between this non-hermetic and hermetic technologies? That insect damage percentage will be as low as 2% in hermetic bags if you store, and seed losses will be like minimum less than 1%. But at the same time, the other bags like gunny bags and all, since holes or crevices are there, like pores are there in the storage bags, so insects can come from outside also into the bag, and from the if the seed lot is infested, the population can move from inside of the bag to the nearby bags also. That is how the infestation slowly spreads in your godown. So important thing is sun drying. So the seed moisture content has to be brought up below 10%. And if you have a provision of any oven or like solar dryers or something, you can use that also. And another technology that has been used is the controlled atmospheric storage. So the carbon dioxide concentration in normal air is like 0.03%. So we are using that principle. So here we are using higher concentration of carbon dioxide, or even you can use a higher nitrogen concentration also. So studies have been conducted at different places, even in India and even abroad countries also. So they have come to even in our ACRB program, we have concluded that around carbon dioxide concentration ranging between 40 to 60%, it can give maximum like 95% mortality of the stored grain. We have tried for rice weevil, pulse beetle also. So we can kill a maximum 95% of the insect pest infestation using this controlled atmospheric storage. Another area we can explore is the host plant resistance, and this table is showing some of the resistant cultivars with respect to the pulses against pulse beetle. So see some of the seed parameters like seed coat, whether it is like hard or smooth, it will be having some tuberculations. Maybe it will be very hard for the insect to drill, even for egg laying also. So it will be the seed coat will be having some wavy structure, so that the adults are like it is very difficult for the adults to lay eggs. So these are some of the seed physical parameters, as well as there could be some biochemical parameters like the content of fat, tannin content, total phenol content. Many parameters are there. So these may be contributing to the resistance, innate resistance within the plant itself against this insect pest. So that also we can explore. Another area is biological control, but this is less explored, even though documentations have been done, and even I am also doing. But the only limitation with this biocontrol agents are that most of these biocontrol agents in the sense parasitoids and predators. So coming to this parasitoids, most of these parasitoids are larval parasitoids. And and these insect pests will be having around 4 to 5 larval instars within the seed. And most of these parasitoids have preference for late instars. Like mostly they'll be parasitizing the 4th or 5th instar of this whatever is the insect pest. So what is the issue is that by the time you kill the pulse beetle larva inside, the seed damage would have been done. So as per my opinion, in this case, a potential biocontrol agent could be Uscana species. This is an egg parasitoid. See here, you can see this is the cowpea seed, and this is the egg of pulse beetle. And here you can see this is a fresh egg. That means it is a healthy egg of pulse beetle. And here you can see black colored eggs which has been parasitized by this fellow. So it has killed the egg. So if we can target the egg stage itself, so there you are killing the further development of the insects. But coming to the larval parasitoids, as I said, it will be little difficult. But still, I just wanted to show you a video. So here you can see the big structure is the pulse beetle larva, and over the pulse beetle larva, you can see some small, tiny larva-like structures. That is this parasitoid larva. See, this is a parasitoid. The name is Anisoteromalus calandre. So this is the mummified. So what this parasitoid will do is that it will be sucking the body fluid of this pulse beetle larva, and slowly, like within 4 to 5 days, the larva of this pulse beetle will die, and that is how. And the larva of pulse beetle larva will die, and following, the parasitoid adult will be emerging out of the seed. So, but it is a slow process, just like we say homeopathy and allopathy effect. This is a homeopathy effect, slow killing, but it will not instant. And another area is the essential oils. Lot of literatures are available where people have tried different essential oils against insect pest, even in the field pest as well as the storage insect pest also. And here you can see a part of the study just have mentioned. So we from this this essential oils of these plants are having some chemical components. Mostly it will be toxic to this insect pest. Some are favoring also, but mostly we are focusing on the chemicals which are having some repellent effect, so that we can use that technology again for the management of the storage pest of seeds. So finally, ending with the chemical management measures. And some chemicals have been already evaluated under laboratory conditions, but a problem with the seed treatment is that we don't have any chemicals, label claim chemicals, for the management of seed infesting pest. But some fungicides and all, it is available under the insecticide, with the insecticide board. But for storage pest, it is very less. And so we have the ultimate solution is the aluminum phosphate tablet, and that also has restricted use. So it has to be farmers cannot just buy that. There is, but still people somehow get the tablets. So we have the recommended doses, how much we have to use, and all those things. So that is one limitation. So we have to come out with some of the insecticides, and even some of the private companies have to come forward, because they always focus on areas which are more profit oriented for them. So I don't know why they have less interest towards the seed sector. So that is why we have very less insecticides for the management of the seed infesting pest. So this is just again the details of this aluminum phosphide and some of the fumigants like methyl bromide. What is the dosage and what is the waiting period available after we use these chemicals. Just before ending, just few areas I would like to. So all these things we already know. So now I just want to throw a light on what are the new areas we can focus. Like you youngsters can focus on some of these methods for the management of the seed infesting pest. So some of the areas are like nano formulations. So it could be a volatile based or essential oil based or a pheromone based. Then another terminology you might have heard is cold plasma. And the third area we can use is the odorant receptor or odorant binding protein. That is actually, these are the part of the insect antenna which we can manipulate for the management of insect pests. So here first, I would like to give you an idea about this. What are the volatile compounds and what is their role? So volatile compounds, it could be used for intra-communication as well as inter-communication. And apart from that, there is another phase where our seed will come, seed, insect pest, and the natural enemy. So it is a tritrophic interaction. So what are the volatile compounds that actually help for this communication? Just, yeah, I would like to say. So, like I told, plant essential oils, it can be used as repellents. And these essential oils, they are having different properties, like it will be fumigant action, it will be having contact toxicity, sometimes it will be repelling the insects. So how we can use is that if the oil is found to be having repellent activity, the egg laying process can be delayed. So that is how we can utilize this technology. And the fourth one is ovipositional deterrence. So it will prevent the insects from laying the eggs. So this is a small preliminary study that we have conducted out our regional station. So 3 plants we have selected is the mentha, that is peppermint, and another one is Lidsia cubeba, it's a Chinese plant, and the third one is star anise seeds. So what we have done is we have extracted the essential oils, that is using the hydrodistillation method, and that oil we have used for the study. So this is the 4 different kinds of test or assays we have to do. So first one is the fumigant toxicity. So for that purpose, this is the kind of vial we are using. When you open this cap, we are keeping the filter paper in a disc shape below the cap, and in that filter paper we will be putting the different doses of this essential oil. And that preliminary dose fixation and all we have to carry out, and finally we will arrive at around 5 to 6 doses. That different doses we have to load into this filter paper into. And after treating the filter paper, we have to keep inside the vial, and then we are releasing the insect pest into this vials. So we have to record the mortality at like 12 hours, 24 hours, and even up to 48 hours also. And in the case of contact toxicity, what we have to do is, this is a filter paper. We have to treat the insect, the essential oil with the filter paper, and then after drying we are releasing the insects, and we have to observe the mortality at 24 as well as the 48 hours after treatment. And coming to this is the we are recording the percent repellents. So here you can see that there is a split. So this is again the filter paper only. First half is controlled, there is no treatment, and here we have treated the specified dose of the essential oil. And after drying, we are releasing the insects. Like here we have used pulse beetle. So we are observing at minutes, like intervals, whether the pulse beetle adults are moving to the controlled disc or to the treated slip. So if all the adults are found sitting on this control arm, means the chemical is repellent. So that is why it is not moving towards the treated arm, treated filter paper. The last one is ovipositional. So what you have to do is just you are treating the seeds with the specified dose of the essential oil, and after drying you are releasing the eggs, releasing the adults, and we will be leaving that for 1 month almost. So if adults have done the egg laying, we will be observing it after 1 to 1.5 months. So if adult emergence is there, means the oil is not that effective. If no adult emergence was observed, means the oil is having toxic effects. That is how we conclude. So just for one example I'm showing, this is the fumigation study we have done for these 3 oils. And I think you all know LC50 value. LC50 means lethal concentration, which is required to cause mortality of 50% of this population. So the funda is like, lower the LC50 value, it means more toxic is your oil. So among these 3 oils, you can see from the table that the lower values were reported in the case of star anise. So among these 3, star anise oil is having the high toxic like fumigant toxicity to this insect pest. So that is how we conclude. Here also you can see the seeds were treated with the essential oil and insects were released. Here this is the control disc petri plate. So here since there was no treatment, you can see the white colored is the egg laying by the pulse beetle, and here it was treated, you can see no egg laying was observed. And simultaneously we have done conducted the germination test also. See, in the control germination was very poor, and in the treated one germination was very good. So that is how you conclude whether the oil is effective or not. So, and you know, many, as I already told you, many oils are there in the market, but the problem with this essential oils is that it is highly costly, and you cannot imagine using large quantity of oils for treating such quintals and quintals of seeds. So you have to come out with the refined technology, like where we can use this nanotechnology part. So whatever is the oil you find to be promising, that oil, either the oil as such, or you can identify that the compounds or the components which are having the insecticidal property. So that you can formulate into a nano encapsulated form, or as it could be a spray also, nano based formulation, or it could be a nano encapsulation also. So whatever is that suitable method you can use, and then come out with the product for the management of the storage pest. To cite one example is that here you can see that in 2022, they have characterized a nanoparticle which was loaded with clove oil. So clove oil at two different concentrations, that is 14.6% as well as 15.2%. Here you can see more than 70% mortality was observed at 14.6% nano formulation, while at 15.2%, almost 90% mortality was cost. So here you can see that the cost reduction will be there. So you are not using the bulk quantity of the oil. Instead, you are either using the clove oil or whatever is the oil in lesser concentration, and sometimes it could be the chemical analog also. So thereby you can reduce the cost of the final product. So this is one area you can explore. Another one is atmospheric cold plasma. So cold plasma is nothing but it is a ionized gas. It will be having like higher concentrations of like ions, electrons, this reactive oxygen species and reactive nitrogen species. So it will be loaded with all these things. So it will be you are using only the atmospheric air, and it will using high voltage dielectric like barrier. One setup is there with maximum voltage. You are breaking down from 220 volts to a lower concentration of around like 80 to 120, and you are subjecting this. This is a non-thermal method. You are using this method to manage the insect pest. So here we can use it in two ways. That is direct exposure of this cold plasma to the different insect stages of stored seeds. Another is indirectly like whatever is a substrate, either you can treat the food or the seed material and then you can release these insects and find out like indirectly whether there is any adverse effect on the development or growth of this insect pest. So just one example I would like to show you is a study has been conducted in 2020 where they have taken we need small containers, and the insect which was selected is tribolium, that is a red floor beetle, and they have taken the different like adult, pupa, larva, all the egg stages, all different stages they have taken. So what they have done is, see, I told you like there is a different direct exposure also and indirect exposure. So for in the case of direct exposure, suppose with respect to eggs or the small, the initial larval stages, what they have done is they have taken a black tape, a sticky tape, and they have pasted the eggs or the larva onto this tape. And then they have kept this tape inside the container, and that container was again covered with a polypropylene cover, and it was exposed to cold plasma. So all these reactive oxygen species as well as reactive nitrogen species, they will stay inside this confined structure. And the impact of this cold plasma, you can observe it. So this treatment period will be around 0 to 20 minutes. And after treatment also, we say PTRT, that is post-treatment retention time. After that also, there were two treatments like the 0 hour, like immediately you are observing the mortality. And in another case, they have kept for another 24 hours or 1 day, and they are recording the mortality. So what they observed is that the first two figures you can see here, this is at a 0 hour post-treatment retention time, and this is 24 hour. So these are the different stages. Like I told you, X stages were taken, young larva, old larva, pupa, adult, different stages were taken. Down is the treatment time. So at the 0th time, there was merely no mortality. Only few eggs they have observed that they have died. In the coming to the next time, that is 0.16, or almost like 80% of the eggs died. But as the time was increasing, the mortality percentage also was increasing. And here also similar trend was observed. And what you are doing in indirect mode, you are not exposing the cold plasma directly on the insect stages. Instead, either the media in the sense the seed or the food material, or that tape I told you, the tape material will be treated, will be subjected to this cold plasma, and then you are keeping the insect stages on track. And then you will observe the same methodology will be followed. And here you will be testing indirectly. So what are there are some biochemical assays we have to do. Those assays like GST, catalase activity, what was the protein content, all these parameters will be analyzed, and through that we can make it out whether the impact of cold plasma has been there or not. So that is all about the cold plasma. Next one is volatile biomarker. Just one second. बंद करने के लिए बोलो 5-10 मिनट. So next area we can explore is like the volatile biomarkers. Volatile biomarkers means these are the volatiles from the seeds. So you all know that seeds will also release the volatile compounds. That is how, see, your most of the field pest is found in the field itself, because the crop is there. The insects are also here and there. So when the new crop is grown, the insects will be attracted to the particular crop. But you just imagine about the storage pest. See, most of the seeds will be stored inside a confined structure, mostly a godown or a closed structure, and it will be dark also. So how come these insect pests are getting attracted to these seeds? So that is actually, you have to think. That is true. These volatiles or the aroma or the odor chemicals which are released by these seeds, our insects will detect that, and they will be following that smell, and they'll be coming into the seed lot. So a small study was done by Tian and his co-workers in 2023, and here they have taken brown rice. So there are different kinds of rice, but for this study, brown rice was selected, and they have tried to characterize what are the volatiles being emanated from this seeds. For this, we need a small setup. We call it as SPME. I had earlier mentioned in my presentation. So SPME is a solid phase micro extraction. It's just simply, I'll explain in very simple terms. So here it is, there is a container. So here you have to keep the seeds. And this is like a syringe. There is a needle structure. So that needle will be inserted into that. We call it as SPME fiber. And this, it is not simply a needle. It is having some adsorbent chemicals are there, depending upon like polar, non-polar, organic, inorganic. So it is available in the market. So you have to insert this needle into this container. Seeds are there. You have to keep like that structure for around like 4 hours. Sometimes it could be 6 hours, depending upon the kind of seed. You have to manage the timing. And after that, so during this time or the period, all the seed volatiles released from the seeds will be adsorbed into this needle. This is the needle, and this needle you have to then inject into the GCMS port. So from there we can get the GCMS profile. What are the volatile compounds or what are the chemicals which is present in this seed? So that is how you get you can document all these compounds. So in their study, they have done this kind of work, and they got around 48 volatile compounds, of which 7 alcohols, aldehydes, ketones, various compounds were there. So here the study was like one set was having set of like rice weevil infested seeds, and the other set was controlled without any insect infestation, and they have like taken the volatile composition at a different storage periods of 2 weeks, 4 weeks, 6 weeks, 8 weeks, and 10 weeks. So they have found that the initial period in the infested one, the alcohol content was very less, but as the storage period was increasing, in the infested seeds, the alcohol content was increasing. So that is how you can make it out whether the infestation is there in the seeds or not. So here you can see the picture. The top side is the control seed, and this is the infested one. So there is a color difference. You can make it out, like some of the compounds are more as the storage period was increasing. See, this is the case of 10 week, you can see more chemicals were reported during this period. And here, this is the PCA score plot analysis done, and they have classified these chemicals into 2 groups based on the infested seed lot as well as the non-infested seed lot. So what you can make out of this is that this is just a single study. Like this, if you have the maximum kind of rice seeds, you have to collected across our country, and you have to document the volatile components of these seeds. So you will have a library kind of structure. So even when somebody sends the seed samples, so you can just match with this. So you have the data of uninfested seeds are having this many volatile compounds. Infested seeds will be having these many volatile compounds. From that library, you can make it out indirectly that this seed lot is also infested or not. So that is the utility of this program. Another just one small study is that, as I told you, there are some of the natural enemies like predators and parasitoids. So I told you, like the insects are getting attracted, storage insects gets attracted to the seeds which has been stored somewhere. How do these natural enemies come to know that these insect pests are there inside the storage? So that is how that's a small study they have done with respect to the red floor beetle and the saw-toothed beetle. So red floor beetle, as I told you previously, there is high levels of resistance being reported, and so very difficult person to manage. And in this study, they have reported a predator, Xylocoris, and actually it's a bug. Bug means it will suck the body fluids of these insects. So for this study, what they have done is 3 categories were there. One was only wheat order. The second was only this natural enemy or the predator order. And the third was both wheat volatiles plus the natural enemy volatile. So these were the 3 treatment combinations. For this study, we need a white tube. It's a simple Y-shaped glass tube. Here you can see that in the picture, one arm will be the control arm. Control arm means the free air will be sent. And here the right side, any side you can fix, and the right side one is the order. So suppose here you can keep the wheat order, and from this Y end you are releasing the insect. So suppose you are releasing this red floor beetle adults, slowly it will, so the air will be passed through and the insects will be moving from this side. It will reach the center, and here it will take a decision whether to move to the free air or the order smell. So they have, this is how you confirm whether a particular compound is attractive or repellent. So if it is an attractive compound is kept here, of course the insect will move to the right arm. If it is a repellent chemical kept here, it will move to the free air because it doesn't like the repellent smell. So this is how we call it as white tube olfactometer studies. We confirm the attraction or repellency of a particular chemical compound. So this table, just simply I want to show you one thing. This is with respect to the red floor beetle. Here you can see that on one arm clean air was sent, and the other arm wheat order was sent. So you can see that it always the insect preferred the wheat volatile. Second study, the natural enemy volatiles were present, and here you can see that clean air was sent. So since the natural enemy smell it doesn't like, all the insects were moving to the clean air. And third was where the wheat volatile plus the natural enemy order was present, and here only wheat volatile was present. Since the natural enemy signal was there, so the insects they were avoiding the this side, and they were moving towards the wheat order. So that is how. So what we can use here is that, suppose we come to know what is that repellent chemical which is signaling the insects from coming towards, which is repelling the insect from moving towards the preferred seed. So those compounds we can formulate into particular like a desirable formulation, and then we can go for a spray or kind of a, like a repellent. We can keep it in your storage godown in the future, and then that is how you can protect your seeds from this insect pest. So these are some of the areas where you can explore. And last one is I told that this volatile compounds are being recognized by these insects mainly using the antenna. So this is the insect antenna. On the insect antenna, this odorant molecule will be recognized. There are gaps in this antenna structure. So the odorant molecule will come inside. There will be odorant binding proteins which will bind with this odorant chemicals, and it will be taken further or transported to the odorant receptors. So just simply I'll explain. Just imagine a key and lock mechanism. So we have a target and a ligand. So if the target and ligand is appropriate, it will lock. Yeah, it will get it will bind properly. So that is how the just for imagine when you are keeping a banana bunch in your table at home, correctly this house flies, yeah, the vinegar flies will come and sit on your fruits. So this is how. So the volatiles being released from the banana fruit, it will be slowly entering their antenna. Antenna sensilla will detect that that banana flavor will enter their antennal structure, and so the binding will happen. So that is how it recognize, okay, that banana is kept over there. So the flies will come and get attracted to this banana bunch. So same principle only we can manipulate here. So we can mimic some chemicals which is having, for example, banana smell. So some other compound which is having a deleterious effect, we can mimic the structure and use for the management of the storage pest. So, and last is the RNA interference and CRISPR-Cas technology, and some few studies have been conducted, and especially that in the case of tribodium, but not towards like larger extent. Few isolated studies are there. So in this two aspects are there, like gene silencing as well as gene editing, and I am more towards gene silencing is the practical possibility, but gene editing I don't know how much it is practically possible. So we can target some of the main insect activities. Suppose, for example, this antennal structure or the development stages. All these stages we can identify the gene, and we can do the correct like silencing of that particular function, and that is how you can like cause malformation in that particular insect, and that indirectly you are trying to manage those storage insect pests. So these are some of the like 4 to 5 areas. I think I have just given you an exposure where still possibilities are there to explore, and science has been progressing these days, and I hope like we can come out with the promising alternative management methods for the storage insect pests that is affecting the seeds. Thank you.
Thank you, madam. Thank you for nice presentation, madam. Any questions from participants?
If you have any like queries or anything, you can contact me. I think I try to make it as simple as possible within this stipulated time.
Very good afternoon, ma'am.
Yeah.
Ma'am,
Please.
actually, from madam, actually, we are going through online workshop from 5 days, ma'am. Earlier, we came to know that delta methane is the only chemical used for spraying in seed storage godowns,
Yes,
and malathion and DDVP are banned. Is
Yeah.
there any gaseous pesticide for spraying?
Actually, yesterday Dr. Revana Siddha have not discussed anything. One evaluation is going on. Actually, the result we cannot disclose much. At another ICR institute, it is going on. So unless and until we get the report about the efficacy of that 3 different fumigants they are testing, so we are all waiting for that. So once that report is getting submitted and accepted by the Central Insecticide Board, then we can go for the recommendation. So as of now, there is no other options.
Okay,
So that is
thank you,
why
ma'am.
we are relying on some other, whether we are trying to see any other possibilities are there or not. Because chemical side, it is very difficult, because I told you, like, companies are not at all coming forward for registering for this insect pest. See, even this delta methrin and all, it is all being ad hoc recommendations through different ASARP programs. That is why we are using that. And most of these chemicals, as you told, it has been banned also.
Okay, thank you, ma'am.
Okay. Anything else?
Anybody else?
Madam, I am Dr. Mutraj from CTCRI Tirunelveli, Kerala.
Yes, sir.
Your presentation is very nice about this one, but we are dealing with the tuber crops.
Yeah, yeah.
We are not having any method for this, madam. What is the procedure? Actually, what happened, no, even frankly speaking, all the aspect, including storage and other things also, even handling the material also. Please tell us any vegetative propagating material, any method is there for this one, storage?
Okay.
Because
See,
what
as
happened, previously I was working in Potato Institute. Now I am working in CTCR, Central Tuber Crop Research Institute, Thiruvananthapuram, Kerala. So we are
Yeah, I'm
dealing
from
with the
Kerala.
quality. Yes. We are dealing with the quality planting material, cassava, elephant putiam, then sweet potato, then taro, tania, Chinese potato, like that. So all are vegetable in that vegetative propagating. So we are for handling the things, it is very difficult, madam. As such, is that scientific method, any technique is there for storage, other things, especially this
mostly.
vegetative product.
Yeah, actually, our institute is mostly focusing on seed related, like scientific method. If you say, mostly it is the fungicides being treated for this tuber crop, that I think, sir, you will be knowing. Apart from that, but my personal experience only I'm telling, it's an indigenous technology. You will all be using the treating the combs with the ash.
Okay, that they were doing.
Hello.
Yes, madam.
Actually, some work is going on in our office, so it's very difficult.
Okay, okay, okay.
So that I have personally found very good for protecting these combs, especially we have in Kerala now. So we always do, and that cow dung slurry.
Okay. Okay, thank you.
Yeah.
Any other questions?
Hello. Hello.
Okay, if no questions means anybody else?
Hello. Hello.
I thank Anjita madam for a nice presentation. Thank you, madam, for very informative presentation. Thank you.
Thank you for this opportunity. Thank you.
Thank you.
Thank you, ma'am.
Thank you.